From the bone marrow, we have the thymus. The thymus is a pyramid-shaped organ. It is found immediately beneath the breastbone at the level of the heart or in the anterior superior mediastinum in front of the heart and behind the sternum. It is called thymus because of its shape. It has a thyme leaf shape. The thymus produces thymosin. Thymosin are hormones that stimulate development of antibodies and also involved in thymopoietin to produce protein found in messenger RNA or messenger ribonucleic acid. Let's go now to the morphological characteristics of thymus or the morphological appearance of a thymus. So on the left side of the illustration, we have a leaf-like structure we call the thymus, and it is covered by a capsule. So it is an encapsulated organ. Now, the thymus is divided into several lobules, which we call the thymic lobule, and each division is separated by interlobular septum. Now, the thymus is divided into two parts. We have the cortex, the outer portion towards the capsule, and we have the middle portion, we have the medulla. So in the medulla, take note, there is a structure which is referred to as thymic corpuscles. So take note of that because we will go into that later on in the succeeding slides. Then on the right side is the histological appearance of a thymus. So you will notice the division, no, interlobular septum and the thymic lobule. And the cortex is the darker portion and the lighter portion is the medulla. So again, I would like to emphasize here that some of the dark portion in the cortex contain also a lighter area, which is also very important part of the histological feature of thymus. We have here an illustration of the microscopic appearance of a thymus. So on the left side, you can see several lobules, no, of the thymus, which is separated from each other by a septum. We call the thymic or thymus septum. And the darker area is the cortex, and the middle area is the medulla. So on the right side of the illustration is a larger illustration of the cortex and the medulla. So C stands for cortex and M stands for medulla. So darker area and lighter area. So on the lower portion is the comparison between the cortex and the medulla. So if you will look closer, both have the same structures involved. So both contains lymphocytes. So these are now the details of the cortex and the medulla. So both contains lymphocytes. Only in the cortex, the lymphocytes are closely opposed to each other, giving the darker appearance. And on the medulla, we have the lymphocytes which are set apart from each other, giving rise to a lighter area of the structure. Another illustration of the thymus, on the left side, we have a red color or maroon colored thymus. So what is emphasized here is the dark coloring of the cortex and the lighter coloring of the medulla. Now, I would like to emphasize on the lighter portion of the thymus, which is illustrated on the right side of the slide. So, in the lighter portion of the thymus, we can see structures that are found in the medulla, which will help us identify the tissue that we are studying under the microscope is of the thymus gland. So we have this structure wherein there is a box, no? A blue colored outline of the box, which are the Hassal corpuscle or a Hassal corpuscle. There are several of them. We have Hassal corpuscles or Hassal bodies. So the Hassal corpuscles is one of the small bodies in the medulla with granular cells at the center surrounded by concentric layers of modified epithelial cells. These are referred to as the thymic corpuscles or Hassal bodies. Now, this was described by Arthur Hill Hassal, a British physician and chemist in 1846. So this structure is credited to him. We call them as Hassal or Hassall's corpuscles or Hassall's bodies or thymic corpuscles. The description of the Hassal body or thymus corpuscle or thymic corpuscle, it is a concentric layer of spherically shaped cells or structures made up of modified epithelial reticular cells in different stages of degeneration and hyalinization. Now, it has an acidophilic background. So there is a pinkish or maybe orange background which is acidophilic in nature against a basophilic background, which are the granular appearance of lymphocytes. So in summary, we have five histological characteristics of the thymus gland. So number one, we have presence of dense fibrous connective tissue which thin out into a loose connective tissue forming trabeculae. So these are now the dividing structure of the thymic lobules. We call the septum. Okay, then this septum will divide the thymus into lobes and lobules, so lobes larger and lobules smaller, which appear to be related and continuous with one another. Second, the presence of dense peripheral cortex with closely packed thymocytes or lymphocytes, and it represents the blood-thymus barrier. Number three, presence of medulla with thymocytes arranged in the diffuse manner. So the lighter appearance of the medulla is due to the loose arrangement of the thymocytes or the lymphocytes. Then number four, which is very important, presence of thymic corpuscles or Hassal bodies in the medulla. So if we will summarize the types of cell present in a thymus, we have the thymocytes, which are the lymphocytes, we have the epithelial reticular cells, and we have the macrophages. Let's go into the secondary lymphoid organs, which are encapsulated. So we have the lymph node and the spleen. So in the illustration, we can see the spleen, which is purple in color or violet in color, and the green outline of the lymph node that is in tandem with the circulatory system. We can see the lymph node. No? So of the two, let's take up first the lymph node. The lymph nodes are small bean-shaped glands distributed throughout the body as part of the lymphatic system. They are small as the head of a pin or as large as an olive, either 1 or 2 centimeters. The lymph nodes may be found singly or in groups, so they could be found in the neck as a group of structure of lymph nodes, in the underarms, and in the groin. And their main function is to filter the lymphatic fluid or the lymph fluid from any foreign substances that may be present. So we have here an illustration of the lymph nodes. So from the breast area to the underarm and to the abdominal area, we have these vessels and we have these small structures or nodules. And these are the lymph nodes. So we have here another illustration of the lymph system or the lymphatic system and immune system. So the green colored vessels are the lymph vessels. And from these vessels are found the lymph nodes. So the lymph node could appear singly or in groups. So around the neck, underarm, and the groin area. Also found in these illustrations are the lymph nodes found also in the abdominal area, and also some of the organs that are involved in the immune system of the body. We have the spleen and we have the thymus gland. Okay, so the lymph node will appear singly or in groups, as mentioned in the neck area, underarm, and groin area. We have another slide wherein it is illustrated the interrelationship between the blood and lymph vascular system. So on the left part of the slide, we have the blue vessel, which are the veins, and on the other side, we have the red vessels, which are the arteries. So smaller vessels for the veins, we have the venule, and arteriole for the arteries. And between them are smaller vessels, both the lymph capillary and also the blood capillary. So there is a close interrelationship between the capillaries. So on the right side is a larger illustration of a lymph capillary. So you can see that the lymph capillary is made up of the same type of tissue as found in the blood capillaries. It is lined with a single layer of squamous cells, or we call the endothelium. And found within the blood capillaries are the different bloods, and in the lymph capillaries will be the lymphocytes. So because of the thin lining in the capillaries, blood cells or the lymphocytes in the lymph vessels can leak out of the capillaries. So they could be found in the environment or outside the vessel, or may be found in the blood vascular system. We have here an illustration of the lymph node. So this is also an encapsulated structure. So there is a capsule present in the lymph node. And there are two vessels that are associated. We have the efferent lymphatic vessels and the efferent lymphatic vessels. So the lymph fluid will pass into the lymph node, passing through the afferent lymphatic vessels, and the fluid will be filtered within the lymph node and will pass out into the efferent lymphatic vessel. So, as mentioned, the lymph node is a bean-shaped structure. So, at the center of the lymph node is a depression we call the hilus, or in some sources, it is also referred to as hilum. And if we will cut the lymph node longitudinally, we can see also the different division inside, wherein the structure which divide the lobes inside the lymph nodes as trabecula, similar to that of the septum. Now, after the capsule, we have a space from the capsule towards the lobe inside the lymph node we call the subcapsular sinus. So it is a space wherein the fluid or lymph fluid will pass through. And we can also see that in the different lobes are smaller lobes wherein we can see the darker area and the lighter area at the center. So the same illustration as in the thymus. So the lighter area is found towards the center, we call the medulla, and the darker area towards the cortex, okay, we have the dark staining area. Okay. Then in the medulla will be some spaces we call medullary sinus for the lymph fluid to pass through, and some of them are arranged into medullary cord. We have here the actual morphology of the lymph node under the microscope. So we can see here the capsule, the different division wherein there is a trabeculae or the septum which divide the different lobes within the lymph node, and a darker area we call the cortex and a lighter area we call the medulla. So in the spaces in the medulla, it could be arranged into cords like a vein, no? Like a vein. And I would like to emphasize here the presence of paracortex. So we have the darker area, the cortex, and we have a lighter, darker area we call the paracortex. And at the center of the lobe, we have a lighter area similar to that of the medulla we call the germinal center. So the appearance of this lymph node is similar to that of the thymus. We have here another illustration of the appearance of the lymph node under the microscope. What is emphasized in this illustration are the presence of lighter area in the lymph node nodule. So we have here a darker area, and at the center we have a lighter area we call the germinal center. And there are arrows that are presented here wherein it is the direction of the flow of the lymph fluid within the lymph node from the body of the lymph node going into the efferent vessel and out of the lymph node. Let's go into the lymph node composition. So, there is a capsule present made up of fibroelastic tissue. So, collagen fibers and elastic fibers. Then we have an outer area, the darker staining area we call the cortex, which is the domain of B lymphocytes, and the middle area in between the capsule and the cortex, we have the paracortex domain of T lymphocytes. The inner area or the lighter staining area of the lymph node could be arranged into cords and sinuses wherein the macrophages and plasma cells are present, and there is a depression at the center we call the hilus or the hilum, which is the mid depression of the node, and two vessels associated. We have the afferent vessels and we have the efferent vessels. The cortex is divided into primary lymphoid nodule and secondary lymphoid nodules. So the primary lymphoid nodules are spherical or ovoid areas of tightly packed small lymphocytes. So this composes the darker area of the cortex. Then we have the secondary lymph nodule, which is a lighter area we call the germinal center. This is a paler central zone with large lymphocytes and macrophages. So in the primary lymphoid nodule, we have small lymphocytes, and in the germinal center, we have larger or large lymphocytes. There is also in the cortex an area wherein we call diffuse lymphoid tissue. This is an internodular cortex and the inner or deep cortex, which are devoid of germinal centers. So we cannot see lighter area at the center. Then the paler inner area of the lymph node we call the medulla. It consists of many lymphatic vessels and medullary cords and sinuses occupied by lymphocytes, macrophages, and plasma cells. So, differentiation between cortex and medulla. Using the low power objective, the cortex is densely stained area or darkly stained area, and the medulla is the pale stained area. Now, the staining arrangement or capacity of the cortex and the medulla is due to differences in the number, diameter, and arrangement of the lymph sinuses and the concentration of lymphocytes, more in the cortex, less in the medulla. The vascular components of the lymph nodes, we have the afferent vessels and the efferent lymph vessels. The afferent vessels are continuous with the subcapsular sinus, and the subcapsular sinus is a narrow lymph-filled space immediately beneath the capsule, and we call them the cortical sinuses, and in the medulla we call them the medullary sinuses, then followed by efferent lymph vessels. Again, we have here an illustration of the lymph nodule, or we refer to them as lymph follicles. So we have here the outer cortex, the dark staining area, domain of the B lymphocytes with a light staining area we call the germinal center. So take note, the B lymphocytes is found in the darker staining area of the lymph nodules or lymph follicles. From the outer cortex, we have the inner cortex, mostly T lymphocytes are found here as seen in the illustration. Again, the medulla, the lighter staining area, is composed of medullary cords and medullary sinuses, and in here are the presence of lymphocytes which are separated from each other, giving to the lighter staining capacity of the structure.